So hi everyone. Good morning, good afternoon, good evening, depending where you are. But welcome to the final webinar series of our International Society for Biofabrication for 2020. We are still running this for 2021, but today we're having our last talk for this year, and we have Professor Tim Woodfield from the University of Otago, Christchurch, who currently is a professor in orthopedic surgery and musculoskeletal medicine. And Tim is also currently the president-elect of the International Society for Biofabrication. Just a little bit of housekeeping rules. If you have any questions, please type it into the chat. The session is hosted and moderated by myself. I'm Kun Lim from the University of Otago as well. We have Dr. Ricardo Lovato from the University Medical Center Utrecht. Then we have Dr. Shrek Zhang from Harvard Medical School and Dr. Andrew Daly from University of Pennsylvania. So if you have any questions, put it into the chat box and we will take the question Q&A at the end of Tim's talk. So without further ado, Tim, I'll pass it on to you to start your session. Thanks.
Thanks very much, Kun, and Shrike, Andy, and Ricardo. It's great to be able to speak to you all today and finish up this webinar series for 2020. You'll notice on the first slide, we're just trying to have a brief introduction to the ISBF for those of you who aren't aware. It's the largest biofabrication society. It's obviously international, and there's a number of great benefits in terms of being involved. One of them, of course, is this fabulous webinar series. So most of these webinars are recorded, and you can, as members, you can access and a lot of content online. So just to cover off a few things around the society is that the membership is relatively cost effective, and again, in the absence of people being able to try and travel to lots of conferences, then yeah, you might want to consider becoming a member. You get access to IOP Biofabrication Journal as a member, and there'll be other discounts you can also get as a member to ISBF conferences, access to then different types of awards. So it's a really great society. Thoroughly recommend you take a look, have a look at the website here for more information, and consider signing up if you aren't already a member. Okay, so I will move on to the talk today. So I'm going to be talking about our group's experience around the development of different bioinks, designing different bioinks and bioassembly platforms for multiple different biofabrication technologies. So I just wanted to start at the beginning a little bit around some fundamentals. And so one of the fundamentals, I guess, in evolutionary and developmental biology of living tissues is that the structure of those tissues or the form of those tissue really follows their function. So you might think of bone and Wolff's law, and so bone responds then, of course, to loading. And so very much the structure of bone follows very much from its function. So again, if you start loading bone, then it will lay down more bone. Again, the structure of the cancellous architecture, is dictated very much by the function of that tissue. Likewise, there's many other different examples. For example, in terms of the structure of cartilage and the development of all of those tissues is being driven evolutionary in terms of an evolutionary process via the function. So if we flip that on its head and we want to then really consider what we're trying to achieve around tissue engineering and regenerative medicine, where we're trying to, I guess, recreate or repair, replace damaged or diseased tissues, we are really trying to engineer, these very, very complex tissues. And so the big question, from this side of things is how do we then go from structure to function? How can we get the right structure that we can recreate that's happened, of course, throughout developmental biology and evolution over many, many hundreds and thousands of years to achieve function. And that's really where biofabrication steps in, because again, that's obviously a technology which allows us to control cellular organization at a very high resolution to try and achieve the types of structures. So that's one of the major goals, I guess, within biofabrication, is to try and achieve these complex structures. So my talk today is going to really sort of cover off two key areas to address sort of some of these different challenges in biofabrication. So we'll talk a little bit about our group's experience around designing functional bioinks, or trying to move towards the development of functional bioinks, which control the cellular niche. So how can we have an instructive microenvironment? That means trying to look at different platforms that allow us or different bioink platforms that can be fabricated across a range of different types of biofabrication technologies, not just extrusion printing. And some tips on how we can control then not only that they're printable, but also that they can provide some cell instructive capabilities for multiple different cell types. The second part, we'll talk a bit more around how we can use different hybrid biofabrication strategies using bottom-up assembly, modular assembly for controlling, also looking at probing some of these different structure-function relationships. Because we sit in a musculoskeletal department in space, most of our focus is in the musculoskeletal tissue engineering, regenerative medicine space. So many of the examples you'll see today around cartilage, bone, and also vascular engineering. So before we jump in too far, and I quite like to cover this again just as part of this webinar, and also because of ISBF, some of the other speakers throughout the year have mentioned also these clear definitions, but I think it's always quite nice just as a reminder that because it's a relatively new field by biofabrication, it's important that we also try and address some of the different clear definitions around where the technology sits. So some years ago now, we attempted to develop a number of different position papers, which were trying to define very clearly the definitions of different terminology within the field. So the point we were trying to do in this slide was just that we were trying to clearly identify where biofabrication sits within wider tissue engineering and regenerative medicine strategies, and to try and explain steps, how we can go from biofabrication to then matured constructs, and make it clear what the different steps are. So in very much general terms, biofabrication can be defined as the automated generation of biologically functional products. So, and that all requires the structural organization of living cells, bioactive molecules, biomaterials, cell aggregates. And that's by using bioprinting or bioassembly. And it also includes the subsequent tissue maturation. So I guess the clear step between classical tissue engineering is that the biofabrication must also include then the hierarchical or spatial organization, so some sort of automation component to it, as well as the biology. That's separate from essentially being able to fabricate a scaffold, which you would then place cells on or post-seed, which would essentially constitute more of a classical tissue engineering regenerative medicine approach. We've done some other further definitions just around to try and tease out some of the further details around the development of or the definitions of bioinks versus biomaterial inks, and their relationship then to the broad range of different types of biofabrication technologies we now have. So obviously the field has evolved very rapidly. We have a very diverse toolbox for the micrometer scale deposition of cells and cell-laden bioink. So of course, the cells being the mandatory component of bioink, or at least the biological components, which can be then combined with an automated processing component, which would define a bioink. Obviously, the range of different degradable biomaterials and also sacrificial biomaterial inks that you can potentially try and use, which would constitute a biomaterial ink. But like I say again, if it doesn't include a cell or a biological component, then generally by definition, it wouldn't be considered a bioink. The point of this overall slide is also just to try and clearly illustrate that biofabrication clearly sits under a wider range of additive manufacturing technologies. So the blue sort of line here is or the blue colors are indicating a range of different light-based technologies, extrusion-based technologies, or jetting-based technologies, which cover additive manufacturing. And the orange-based ones are then now these rapidly advancing subsets of biofabrication. Again, with the idea is that these biofabrication technologies are now incorporating the use of bioinks or multiple bioinks in combination, then potentially with different biomaterial inks, to then generate complex cell-laden constructs or cell-instructive constructs. It also includes a couple of examples here where we're now seeing the rapid emergence of different hybrid biofabrication technologies. So these are combinations then of multiple bioinks or multi-component bioinks. Again, different assembly technologies, combinations of different additive manufactured technologies such as melt electrowriting or extrusion of thermoplastic materials in combination to then provide a whole additional advancement and really push the boundaries in terms of the levels of complexity of tissues we can try and fabricate. So under that, I'd like to sort of go back and just briefly show a rather historical slide of some of the work we've done previously, which is very much a classical tissue engineering strategy. And this is some work we did back some years ago. We were trying to look at fabricating mechanically stable constructs using anatomical matching of in this case was a rabbit knee. So we're able to take some CT images. We're able to use the capability of in this case melt extrusion of a thermoplastic polymer in a range of different properties, which we were then able to try and then post-seed. So essentially we were trying to look at the macro scale anatomic structure here that was porous with the right mechanical properties to match articular cartilage, and we were then able to take autologous chondrocytes from the rabbit and look at seeding and tissue formation over time. So I think this was again one of the first studies we were actually trying to look at articulation between two tissue engineered constructs that have been fabricated using melt extrusion and seeding of autologous chondrocytes, with the idea of looking at regenerating or surface joint resurfacing for cartilage repair. It went back to some of the very beginning work we'd ever done around demonstrating a capability that we could melt extrude thermoplastic polymers, and we were very much targeting that the structure and function of those materials then match the dynamic stiffness of articular cartilage, and that when we also formed tissue that over 28 days in vivo that we could get even an improvement in mechanical properties. So we're trying to essentially make sure that the structural component of these scaffolds had an anatomical shape, were porous, and were promoting the best possible microenvironment that when we placed cells within that microenvironment, we could get differentiation. So in this particular case, we're able to try and show that prior to seeding, we had very good hyaline-like cartilage, which we're able to try and tissue engineer. We're able to then place it into the joint. We're able to try and show that the subchondral bone regions were able to be populated with bone, and we got excellent fixation over time. Unfortunately, well, not unfortunately, of course, it's an extremely aggressive environment. You can imagine in terms of us being able to perform a osteotomy of the femoral component in this in vivo model and replace it essentially with a fully engineered construct. The remodeling process meant that we lost some of the hyaline cartilage that we had originally engineered. We still were able to maintain an articular surface, but the reason that I'm trying to give this example is that that was sort of the latest, I guess the latest technology at the time to be able to try and have an anatomical structure deliver autologous cells that have been pre-engineered to try and look at joint resurfacing. And again, it's an extremely, like I said, aggressive environment. And despite having the anatomical features and also the mechanical support, the remodeling processes meant that we weren't able to maintain that high level of hyaline cartilage. And while we still had articulating surfaces, it had remodeled in a number of different regions to more of a fibro cartilage. And so we need more than potentially just cells and these thermoplastic materials to be able to then engineer functional tissues. It may be over time, again, this could have remodeled into hyaline cartilage again. And to be fair, we did essentially still were able to maintain function even though we didn't have the perfect tissue quality. And so I think that also raises one really nice question, is how perfect does structures have to be in order to be able to achieve, really good function long term. But what I'd like to try and focus on is what can we do now if we're of course trying to provide additional capabilities in the form of hydrogel-based bioinks. What can we try and do to enhance that cell instructive capability to make sure that we've then got different hybrid technologies to be able to try and deliver on some of the different challenges we have in regenerative medicine. So one toolbox we have, and this is what we're going to try and focus on a little bit in this talk, and obviously in our group, is around being able to have tunability and control of our different bioinks. And the major way we do it in our group anyway is around looking at different photo cross-linking technologies. We recently just reviewed the sort of fundamentals in the field with Jason Burdick, and so I'd highly recommend you have a look at that paper just to be able to go through all the different technologies, because I'm not going to mention all of them at all today, but I just wanted to focus on a couple of different technologies, primarily what we're doing around photo cross-linking for traditional extrusion bioprinting, as well as then lithography-based digital light processing. So here are two examples of two different biofabrication technologies: extrusion 3D bioprinting and lithography 3D bioprinting. And I wanted to put this example up because, as you will have potentially seen in a number of different webinars throughout this series, is the challenges that we have in terms of moving or developing bioinks for each individual biofabrication platform. So many of you will be very familiar with extrusion 3D bioprinting. Essentially, when we want bioinks for extrusion bioprinting, we want them to be able to try and flow out of nozzle. However, they need to be viscous enough, either in terms of having formed a gel in the syringe, that they can then also rapidly recover. So in a lot of cases, we have a phenomenon that we're trying to achieve around shear thinning. So essentially, we would have capability that under force or under shear, our hydrogel is able to then flow, so it can flow through the nozzle, but then it has shear recovery immediately upon removal or of that shear force upon exiting the nozzle, so that essentially you're allowed to have some sort of gel that can be self-supporting, and so you don't essentially end up with a puddle. Of course, we would like with biofabrication to print complex constructs, and so the shear thinning capability of viscous bioinks is essentially one of the major criteria that we require, that's needed in terms of printability. Normally, once of course you've got that shape, you can post cross-link it. Often that's done with light, and there's a number of different strategies in there. So essentially, those are the requirements you would need for extrusion 3D bioprinting and your bioinks. With lithography 3D bioprinting, it's a different type of process. Essentially, in this case, you have what you would like to have as a very non-viscous bioink. And of course, what you'd like to be able to do is be able to cross-link that. Again, most of these available lithography machines use light. Essentially, you're able to then cross-link regions of light where you project it, and you have a stage which can then allow you to subsequently cross-link multiple layers. And of course, in all of these processes, we'd like to incorporate cells within the step. So the rheological profile is quite different between each of these different systems. So by way of example, if you wanted to then try and come up with a way to be able to have a similar bioink or material that can be used across multiple platforms, then it's extremely challenging because there's a number of different criteria you have to look at. Again, if you're using different types of light systems, you need to carefully consider what photo initiator you're using, what biomaterial, again, what's the rheological profile, and then of course we need to make sure that we can incorporate different cell instructive capabilities as well as the cells, and that those cells remain viable. So there's a number of different considerations that have to be thought through. So I want to introduce our visible light photo initiator system. So this was developed by Kuhn Lim in the group, and just very briefly, it's a ruthenium metal complex system, and it's I guess an interesting alternative then to potentially some of the other more commonly used UV-based photo initiators, including Ergocure. Essentially, the process is under, if you have a ruthenium and sodium persulfate based metal complex system, then under excitation with light in the 400 to 450 nanometer range, you get excitation and oxidation of ruthenium. that then generates sulfate radicals, which can then drive essentially a chain growth polymerization of your hydrogel. In this case, this example here is then gelatin methacryl oil. So that is a mechanism for essentially then some radical-based polymerization. We recognize that again with using visible light, particularly the ruthenium, is extremely high molar absorptivity, and so very low concentrations of the ruthenium photoinitiator can allow quite complete cross-linking. Using visible light, you get quite good penetration depth compared to potentially UV and ergocure. We see some differences in cell viability with some of these, with the ruthenium-based visible light photoinitiators, similar to other visible light ones, including LAP. And we can then also look at downstream tissue formation. And some of the reasons that we think there's benefits there across the range is also, again, we know that UV has the potential to cause DNA damage, particularly at high exposures. And so what we see here also is that, again, that you can have increased reaction oxygen expression with exposure to UV, which we generally don't see, or again is at a lower extent under visible light. So it's an interesting photoinitiator system, which is highly tunable, that we've been investigating for quite some time. Here's some examples where we've used the photoinitiator system to then create bioinks for extrusion bioprinting, including obviously gelatin methacryl oil, where we can then get quite large constructs with quite high shape fidelity, maintenance of shape fidelity, and cell viability. We were obviously very keen to try and see if we could use it for different lithography-based bioprinting. So in this case, we started out with a synthetic system for some of the preliminary work we did using PVA, a small amount of Gelma, and also a photoabsorber. And I'd just like to sort of use this opportunity to show that then the changes in resolution you can get in terms of different biofabrication platforms. So with lithography, we're able to then generate quite complex structures. So like I say, these interwoven features, which you can't easily do using extrusion-based bioprinting. And essentially the different pixel or voxel resolution we could achieve was around down to about 25 microns. So again, this is another sort of higher resolution layer by layer process. And again, with the addition of Gelma, we're able to then allow us to incorporate cells within what we were then terming at this stage of bioresin, because again, it has quite unique features, which essentially would dictate a difference from a standard Bioink in terms of the types of low viscosity properties you would like, including a photoinitiator system. So if you want to find out more, there's a couple of the papers there. But I think one of the interesting things we did in this early work, and this was also done with Ricardo at UMCU, was also to try and show that when we incorporated cells into the process, not only were we able to maintain viability and see differentiation, but what we wanted to show as part of the layer by layer process that we didn't end up with sedimentation. So this actually shows that we could print a relatively thick construct, and then we were trying to look at the cellular distribution throughout that we then actually ended up with a homogeneous distribution of cells throughout that that cell laden bioresin print. So it's quite an attractive way then now to actually print really high resolution parts, incorporating cells that can then actually form or look at differentiating over time, again, albeit in a semi-synthetic initial bioresin formulation. So in terms of where the field has now then gone, some of the latest developments that have popped up very recently, again, are other types of biofabrication systems, one of them being volumetric 3D printing. So you can also then, of course, utilize the capability of projecting light. In this case, it's essentially projecting a volume of light, which allows you to fabricate very large constructs within seconds. As an extension, I guess, of even some of the lithography techniques I showed to you. Essentially, you project on a rotating liquid bath, and then of course exactly where the light shines is then what you're able to try and crosslink. Ricardo and the UMCU team have then also been able to demonstrate that you can incorporate cells into part of that process and the speed with which it can occur. And again, some of the examples that showed up in terms of this initial publication, there was examples using GELMA and the ruthenium SPS visible light photo initiator system. I believe Ricardo's also used LAP. It's also worked as well as part of the ruthenium as a secondary cross-linking process. So just trying to show that there's now some alternative photo initiator systems and their tunability and capability across multiple different platforms. I'm going to do a quick plug here actually for both Koen and Ricardo. They're going to be running if you're interested or working in the area of light or using or harnessing light in biofabrication, then there's a special issue they're trying to put together in the Biofabrication journal. So I just want to change gears slightly, of course. So we've looked at different ways of photo cross-linking and in terms of actually the photo initiator system, and that it can be applicable across multiple different platforms. But again, some of the examples I've shown you haven't necessarily been able to then demonstrate that while they're also printable or can be used across multiple platforms, that the different bioinks are able to then support a cell instructive niche. So again, that might require a range of different capabilities. So in terms of how we would design a three-dimensional hydrogel or a hydrogel microenvironment, is that again, it's a highly hydrated microenvironment, which makes it very favorable for cells. And there's a number of different things you can try and tune or that you can try and focus on. Some of the different examples are all here, particularly in terms of a Bioink. We would like to, of course, look at ways of tailoring the stiffness. That's obviously one way that we can clearly provide cell instructive capability within our hydrogel Bioinks. Look at growth factor presentation or release or different extracellular matrix components, which can then make sure that the cells that we want to try and print are going to be happy and instructive. So this big challenge here is that how can we then look at designing these different types of niche and using hydrogels that are printable. So we can show that we can print certain hydrogels, but are they printable then with the cells that we want, and can we make them cell instructive? So I'm going to present, I mean, there's a number of different examples. I'm going to present a couple of examples of experience within our group. So first of all, in terms of development of essentially an osteogenic bioink, one way is that you can also, to provide cell instructive capability, is to include different differentiation cues. In this case, in the form of strontium and a nanoparticle formulation. So what we would obviously like to be able to do if we're going to look at additives within our bioinks is to, first of all, make sure that we have additives which can modulate the printability. So are they sort of rheology modifying capabilities. So obviously the inclusion of different nanoparticles within a hydrogel can provide modifications in terms of then their viscosity profile. So in this case, again, with a range of different concentrations of strontium, these are strontium sort of nanorods we were able to synthesize. We incorporated those and looking at essentially a GELMA strontium bioink. We're able to get subtle differences, which then can modify the viscosity then at the temperatures that we would like to print at, and show that we still have a shear thinning capability. So again, the viscosity decreases then with increasing shear rate. We came to an optimal strontium concentration and can show that we can then, again, if you just take 5% GELMA, so a relatively low concentration GELMA concentration, you don't end up with nice print fidelity. This shows then obviously the addition of strontium can remedy that, and that we then can also have good cell viability with the inclusion of these cell instructive strontium nanoparticles. Strontium, of course, is a potent osteogenic differentiation factor. So it was nice then also to see if we able to print these constructs in the presence of mesenchymal stromal cells and culture them in the presence of osteogenic factors, we can get mineralization and nodule formation in these inks over time. So that was kind of a dual function capability of using a strontium nanoparticle. So one of the other things we would potentially like to do is add a third thing, is that whether those, whatever our additive is, may also then be able to try and look at recruiting or sequestering different types of growth factors. So this was some work we did with Richard Orifo's group at the University of Southampton. They've been working with the development of different laponite nanoclays. So laponite is a disc-shaped nanoparticle with a positive rim charge and then highly negative surface charge, and can be used to make a number of different colloidal suspensions. So we were curious to know whether it would have a similar rheology modifying capability for the development of a gel-molaponite nanoclay. Also, it's again a potent osteogenic differentiator as well as a growth factor binding, shown to be have a growth factor binding capability. So like before, we were able to come up with an optimal concentration of laponite, which allowed us to have printability, that we can then also show that in the presence of osteogenic differentiation factors, dexamethasone, we can get differentiation. And again, as an interesting side note, in the absence of some of those different osteogenic factors, we can also start to see nodule formation in some of our gel-molaponite bioinks. What we also wanted to do, though, was try and demonstrate in an ex vivo model. So this is a choanteric membrane assay, so a chick egg assay, where we're trying to look at then the sequestering of VEGF and vascular network formation within this particular model using the gel-molaponite constructs. So just very briefly, again, if we could take our gel-molaponite in the presence of VEGF, we could see that we could get vascular invasion, or at least vessel formation within these constructs. Again, in the presence of just straight laponite and gelma, we could also still see some vessel formation, but in the absence of any laponite, then we did not see penetration. So again, it's providing a very permissive microenvironment. So again, that was interesting to be able to try and see that we can now have I guess a multifunctional capability of these gel-molaponite nanoclays. So that provides some examples of different bioinks and bioresin platforms we've been working on. And so far, what I've been trying to, or what I've demonstrated, is some examples of different chain growth mechanisms using a visible light photoinitiator. Again, some different types of factors, then we can try and also then incorporate into our bioinks to give some cell functionality. There are other different polymerization mechanisms and strategies, though, that we can potentially utilize to leverage even further capabilities. So I wanted to talk about those a little bit further. So that can be also around the use of thiolene chemistry and thiolene photoclick chemistry using the same photoinitiator, but relying potentially on alternative ways of using step growth polymerization, which provides a number of advantages over some of the other techniques I've explained so far. And the main thing is, again, if we can have tunable systems, again, that are highly controllable, then there's a possibility we can use those, or that the types of bioinks that we're developing have inherent properties which mean that they can be used for fabrication across a range of different types of capabilities. So like again, we recently reviewed a range of different types of multi-component bioinks, and so I'll point that to this paper here if you're interested to read on a little bit further. So one thing I wanted to try and focus on, of course, is these different platform bioinks and more of this step growth polymerization. So we've been working with allylated gelatin. So again, we've termed this gel age. So again, what we're trying to then rely on as part of this is whether we can look at developing a tunable photoclick and of version of these allylated gelatin, and whether they have inherent properties which mean they can be used across multiple different types of platforms. So again, this essentially relies on in terms of cross-linking the dimerization, of different thiols, again with then different reactive carbon-carbon or ene groups within these hydrogels to generate a very homogeneous and complete cross-linking process. So what we're able to do is work with a family of different types of gel age materials, and what we were discovering when we did quite an extensive range of rheological characterization, and this was done with Jürgen Groll's group at the University of Würzburg, is that at low temperatures we had a really nice viscosity profile which made it ideal for extrusion-based bioprinting in the presence then of a range of different types of cross-linkers, including DTT or PEG, And when we included cells, we saw really nice cell viability. So again, that's trying to use the same photoinitiator system, and we could now have a gel age Bioink. Likewise, when we also then started looking at room temperature, which is generally where all the different lithography-based systems work at room temperature, again with visible light, we showed that we could have quite a favorable rheological profile. So again, low viscosity, which made it ideal as a bioresin. So we could also show with these gel age materials, we could get high resolution DLP structures and good cell viability. So that was a really exciting finding, and that then tried to show the capability of these the sort of photo-click chemistry, and showed us that we could then actually have one family of materials that could start to be used across multiple biofabrication technologies. Could we use the thiolene click chemistry to impart even more advanced cell instructive capabilities? Well, the answer was yes. So previously, Gabrielle Lindberg and my group had been working with a range of different thiolated heparin molecules as a growth factor binding molecule for regulating chondrogenic differentiation. And we were able to show that using, when we were able to then incorporate thiolated heparin, we got excellent retention within the gel age process. Again, the step growth cross-linking process, very efficient with retention of thiolated heparin. Again, the presence of heparin then meant that we could get enhanced tissue formation, and then after longer term cultures, that then resulted in increased mechanical properties. So in terms of chondrogenic differentiation, this is when we're looking at an interesting Bioink, which could control a cell instructive capability for cartilage tissue formation. If we're then looking at how we can tune hydrogel stiffness, again, the flexibility of the process in terms of being able to modify gel age concentration and cross-linker ratios meant that we could actually get a broad range of different mechanical properties from quite low around down to sort of 10 or less than 10 kPa, all the way up to around, say, 250. Which meant that we had quite a nice broad range of different properties we could achieve that were printable, again, which you often can't get with GelMA. The interesting thing is that we could then show that the incorporation of cells, so again, the step growth polymerization, cells didn't necessarily affect then any of the downstream properties or disrupt any of the cross-linking mechanism, again, which we've observed in our hands, at least anyway, quite a reduction then in the properties that you achieve with GelMA in the presence of cells, which of course is what we want. So some quite nice tunability properties. What does that mean in terms of the structures we can try and fabricate? Well, again, for vascular-based bioinks, of course, what we would like is very, very soft, permissive hydrogels to allow network formation. What we know is that, again, you get network formation in soft hydrogels generally below 5 kPa. If you have a soft hydrogel, again, above 20 kPa, you don't end up with a permissive microenvironment. Again, with a co-culture of mesenchymal stromal cells and GFP-labeled Hubex, then what that means is that if you would like to then generate a soft permissive hydrogel, you can't achieve it any sort of print resolution with GelMA because it doesn't have the right rheological profile. But what we can obviously show is that the advantage of gel age is that you can have printable constructs with good shape fidelity that are soft as well as stiff. And so you can start to tailor those microenvironments for what you would really like to achieve. And that's really starting to address some of these sort of novel areas of materials that you would like around, biofabrication windows these days, is that we would like to be having tunable constructs which give you shape fidelity, which is what you obviously want in terms of making sure you can get cells placed wherever you like them in a hierarchical structure, but again, that you've got a relatively soft permissive environment that's cell instructive. So what are some other tricks and tips that we've published more recently? So again, this is led by Koen Lim and Bram Solomon in the group. What if we wanted to push the boundaries even further? So we go to really low weight percent polymer concentration. So in this case, if you take a 3% gel age, for example, it's essentially extremely low viscosity, and you can't print with it. And again, it would be the same if you're taking very, very low concentration, GELMA, and other things. So essentially it's non-printable. But if we think about some of our chemistry and we think about the thiolated cross-linkers that we use as a reducing agent, the sodium persulfate in the photoinitiator as an oxidizing agent, then essentially we do have a redox reaction occurring. And if you leave that running over time, then is there some way that we can change the properties of our bioinks over time? So what this shows is, again, we can get a nice sol-gel transition over time, and that we actually either increase the complex viscosity of these materials due to this redox. So again, this is without any light. We're just purely relying on having redox cross-linking mechanisms running. So that then starts to mean that you can take something that's non-printable, and then over a period of time, it actually allows it to become printable. So again, if you essentially allow the redox reaction, of course, you won't generate a very stable long-term hydrogel, because there's no, you haven't necessarily done any secondary cross-linking. But if you take your essentially primary cross-linked construct, and then you do a secondary cross-linking step, this time with light, using again the ruthenium SPS, then you can end up with very stable constructs, again, where we see in our hands nice cell viability and starting to see migration of again of mesenchymal stromal cells. So again, this is an interesting way of harnessing the capability of the photoinitiator system. Again, this was work done again with collaborators with Jelena Raniak-Kovacina at UNSW in Sydney, and working with Silk, and then also Stephen Tree within the group here. Again, trying to look at then alternative chemistries. So I mentioned before that obviously the ruthenium system, when it's excitated, can generate these sulfate radicals, but also the highly oxidized state with ruthenium 3+ can also then look at other ways of cross-linking. And in this particular case, we're trying to work out, could we utilize this to allow cross-linking of silk fibrin? Again, another alternative different types of hydrogel, which then provides a range of different cell instructive capability that's completely different, obviously, from gelatin based systems. So obviously we're trying to look at then oxidizing these different aromatic residues such as tyrosine within this process. And again, because we have a number of these different tyrosine-rich proteins in silk, we were trying to work out whether we couldn't also then look at cross-linking using dityrosine formation within these native tyrosines that were present within silk. That was compared to generally enzymatic cross-linking, and for the interest of time, I won't go into a whole lot of details and just refer you to the paper. But of course, with silk hydrogels, the main big advantage, of course, is that we can rapidly cross-link these within minutes, whereas the enzymatic cross-linking often takes a very, very long periods of time. So we can rapidly do this, which is very important for cellular incorporation. And we find that if in this case we're doing a 3D bioprinting process using sacrificial printing, we can end up with silk constructs which have high resolution features we can incorporate cells into, and we're trying to take account this capability of these visible light cross-linking mechanisms. One other example also in terms of using similar types of technologies is to say, well, and we don't necessarily just have to only cross-link the hydrogel, is well, we also have a number of tissues we're working with which potentially might also take advantage of this different dityrosine or biphenyl cross-linking. This is just another example. If you're trying to then look at seeing if you could print a hydrogel construct that could then be printable, but then also look at binding with native surrounding native tissue. This is just an example with essentially a GELMA tyramine. Again, work done with Ricardo and Kuhn, where we can try and show that in the presence of light, we can also get what we think is again cross-linking between tyrosine-tyramine groups. So again, by incorporating tyramine, we can then also get cross-linking with native tyrosines within surrounding articular cartilage. So this actually shows two cartilage biopsies that have been adhered together using a single, or I guess a dual functionalization process for cross-linking our hydrogel, but also then binding surrounded native tissues, which show that we get a 15-fold increase in the mechanical properties compared to essentially just trying to look at photo cross-linking GELMA alone. I'm just going to skip ahead. I think obviously there's a broad range of really other exciting techniques for being able to then cross-link low viscosity bioinks or really complex structures. So the Burdick group looked at a number of different ways of looking at in situ cross-linking. There's obviously the fresh technique pioneered by the Feinberg group, which allows you to print very low viscosity in a suspension bath, which you can then release over a period of time to then print really large scale constructs, which are essentially self-supporting and have significant capability to, like I say, print very, very large clinically relevant size features. There's some other really nice work in the lithography space, again, also from the Miller group, again, showing that you can print very high resolution functional features that can be perfused. In this case, again, perfusing oxygen into also a perfused chamber of a vascular network for an alveolar structure. And I think the further advantages or further developments we're going to see in these types of technologies is then, of course, incorporation of cells into these processes, which is a bit of a challenge at this process. And in certain examples, like we showed earlier in my talk around how we can use different bioresins to incorporate into the bulk of these lithography bioinks. So just in the next aspects, I just want to cover off some other aspects around some of these hybrid spheroid fabrication techniques. So obviously there's a number of different ways we've incorporated cell instructive capability into different types of bioinks, but could we actually look at some other completely different biofabrication strategies which also try to provide cell function or cell capabilities to these different types of tissues, or allow us a completely different approach to how we might also address those. And the rationale behind using different cellular spheroid techniques is really the fact that we can try and utilize or leverage different bottom-up assembly processes, and that we can try and use the automated advantages of biofabrication to in combination with these self-assembly or spheroid fabrication technologies, which essentially have rapidly developed over the last 5 to 10 years for moving in vitro screening from 2D to essentially 3D culture models. So there's a role I feel that that biofabrication can play to make sure that we can look at furthering different types of in vitro 3D in vitro tissue models using modular assembly. Just by way of brief introduction, a number of you will be familiar, but there's a range of different ways you can form tissue spheroids. So again, in terms of straight cellular spheroids, you can use different hanging drop methods or different microgravity-based methods for forcing cells into a suspension, and there's different sound-based methods also, which are able to force cells into aggregates. You can also do forced floating or different stirred tank bioreactors for forcing cells into an aggregate format. One other way is that you can also then use biomaterial hydrogels essentially to also aggregate cells, so either in terms of microparticles where cells form on the outside, or you can do cell encapsulated capabilities of hydrogels within those cells. There's a range of different ways you can form these tissue spheroids. And historically in biofabrication, this is where a number of different technologies were pioneered. So, Gabor Dvořák's and things showed, again, that you could allow, or you could use bioprinting to place different spheroids. You could get fusion over time into complex constructs. And there's a number of other different examples over time around how modular tissues could be automated, in an automated way, could be fused over time. To generate a range of different shapes, structures, not necessarily always functional, but were essentially the pioneering work in this area. We recently reviewed those, and here's just some examples also of where we would like to try and take a range of different hybrid biofabrication technologies to make sure that we can assemble a broad range of constructs. So again, this is work that's happened over about the last 15 years around combinations of different hybrid biofabrication as well as additive manufacturing technologies to generate a broad range of different functional constructs incorporating different spheroid technologies. So if you're interested, check that paper. So just to highlight one area or one example where we've automated this process was essentially a bioassembly technique where we looked at combinations of tissue spheroid fabrication and our construct fabrication. Again, what we're trying to rely on here is that we can promote the self-assembly and cell signaling capabilities, which are mimicking then different cellular mechanisms and regenerating this condensation niche you often see in developmental biology. So by able to essentially allowing cells to form or self-assemble into spheroids, replicating that process or making an automated high throughput way, then we can try and recreate different complex tissue structures. So we had a number of different ways of doing that, a 96-well plate format where we could essentially centrifuge cells, allow them to form dense microstructures at large scale, and we then had an automated way to then try and look at assembling essentially hundreds of these different cells or cell co-cultures. This other example that was there was essentially a microfluidic process, which I'll talk about in a minute. The idea is that then if you can automate this process, then you can print a range of different thermoplastic materials and look at bioassembly as well at the same time to create complex constructs. This is our automated process. Essentially, it allows you, this is how we try to assemble different spheroid structures. So to be able to take all of our spheroid structures and then be able to place them within a particular location within our printed construct, we needed essentially a singularization mechanism and injection system. And this essentially shows the automated process where we have spheroids coming in, one spheroid is trapped and then passed down. So we're able to then essentially have an automated way for delivering single spheroids at a time for then placing within a construct. And this is just a video of the process, then trying to show that we can have a broad range of automated systems and that we can then, in a layer by layer process, start to automate the complex assembly of different multi-component tissue spheroids to study then fusion over time. So this is trying to show that we can have complex structures fused over time. Again, this is trying to look at chondrogenic differentiation, and this is fusion and cartilage differentiation, where we can then see tissue formation and tissue growth from these modular assembled constructs. And again, we have maintenance then of different zonal layers of either then different combinations or co-cultures of articular chondrocytes versus mesenchymal stromal cells into essentially what would, in this case, an osteochondral capability. So I'd just like to try and wrap up and I'll just try and zip over some final examples here to try and show where we've ended up at in terms of our high throughput mechanisms. So obviously we can utilize the all of the visible hydrogel and cell instructive capabilities that I've mentioned earlier in the talk around the assembly of different gel age constructs to try and drive cellular differentiation within these cell encapsulated hydrogel materials. Again, we see in these microgel formats that we get excellent tissue formation like we did previously in the different bioinks, and that you can then utilize the advantage of the mechanical properties that you see, again, to make then microgels that can have a vascular network formation or vascular niche based obviously on being able to have very, very soft, tunable mechanical properties, which allow you to then have network formation and vessel growth over time as you end up with maturation, again, in this case with an MSC HUVEC co-culture model. So I'd just like to finish there with sort of a summary of where our work has gone and where some of the future work is going. Of course, what we would like to be able to try and do is develop complex models for screening, and that we would like to obviously have mechanically stable hybrid fabrication technologies. Again, we can try and then tailor the mechanical properties to meet the target tissue that we'd like. In this case, this was an example for cartilage. We were then able to place a range of different types of modules. We've shown that those modules can then fuse and mature over time by having the right cell instructive niche, and that each of these then modules can incorporate a range of different types of target tissues. So again, in terms of potentially having bioinks that can support achondrogenic differentiation, vascular network formation, or osteogenic differentiation. As we move forward, the automated capability of all of this means that we essentially can have formats where we can have biomaterial spheroids, where in those particular modules we can control certain microenvironments around oxygen or cell density. We can then have more complex biomaterial-based microgels, where there's a broad range of different types of aspects we can start to tune and control. The capability of this means that we can then have a very I guess a very sophisticated toolbox even further to be able to then probe different structure function relationships. So what do all of these factors mean when we then start to try and look at more of an organ scale? So rather than necessarily looking at screening of individual spheroids or organoids, can we use the capabilities then of different bioassembly techniques to generate in vitro models for high throughput screening of multiple spheroids or organoids, multiple cell types, to really start to probe some of these different types of structure function relationships. I've shown some examples primarily around cartilage, bone, and vascular networks, so musculoskeletal, but we feel this is quite powerful in terms of where it might translate over into different organoid biology work, and see a really important role that biofabrication has in addressing that. So with that, I have a slide, sorry, with different acknowledgements that I just wanted to jump to. This one, just to really acknowledge all of the great work from my team here in the CREATE group at University of Otago, and our fantastic collaborators and funders, and would like to invite you all to the next ISBF annual conference in which will be in September 2021, as a virtual meeting, as well as a potential face-to-face meeting in Wollongong in Australia. Thanks very much, and happy to answer any questions you might have.
Thank you, Tim, for a great talk. Some really nice images and videos throughout. So we have a couple of questions from the audience that we can go through quickly. So the first one is related to the strontium, and what are the advantages of strontium over hydroxyapatite, maybe?
Oh, sure. I mean, good question. We chose strontium just in terms of the form factor. So again, in terms of being able to have a nanoparticle where we can look at rheological modification, that was one of the key reasons. Certainly, there are a number of examples where people have used hydroxyapatite. I guess with a nanoparticle, you can get a very high surface to volume ratio. So in terms of then the distribution, then within your Bioink formulation, then you can get quite a nice homogeneous distribution. Again, being able to have that particular type of nanorod structure we found was quite favorable. So again, essentially just meant that we could have very high surface to volume ratio or surface contact ratio with that particular format. But that was just one example, and certainly hydroxyapatite particles can also be used. The question is again whether you would see the same rheology modifying capability, again depending a bit on the form factor of what that particle was. But yeah, it would certainly also impart some strong osteogenic differentiation capability.
I guess there's another question in this part of the talk. And what kind of mechanical stiffnesses are you trying to achieve for the bone in vitro models from the bioinks? And I guess, are they kind of matching native bone, or
Yeah, that's a good question. Obviously we don't try to target hydrogels. Obviously hydrogels and hydrogel bioinks are always a challenge to be able to then actually get mechanical properties similar to bone. So It goes along the lines of that bioassembly strategy I was presenting at the end there. The idea is if we can provide the right niche along with the other combination of hybrid properties, then or hybrid biofabrication technologies, then that's where we see some good strong advantages. In particular, that we can also potentially have vascular modules that can help drive the drive essentially vascularized bone at the same time. So again, whether you have an ink that's extrusion bioprinted, or potentially you'd have vascular modules that are forming a network in combination then with a bone or osteogenic Bioink or modules. There's a couple of different strategies in there, but yeah, we certainly it's certainly a big challenge. We wouldn't be targeting mechanical properties in the osteogenic Bioinks that would match bone. We would then be looking at a range of different other nanocomposites. We have included strontium nanocomposites along with other different thermoplastic polymers. There's a broad range of really nice examples out there of also then additive manufacturing or bioprinting of nanocomposite materials that have properties that are starting to approximate native bone. Again, the question is how cell instructive are they, or do we need these combinations then of the cell instructive niche along with those really strong mechanical properties, and that's where I think we'll always need a combination, a bit of both.
Ricardo, do you want to take over?
Yes, there was also another question from Ernouche Raffier. How can you overcome disadvantages of light cross-linking precursors in cells? So I guess the photoinitiators and their biocompatibility.
Sure.
Can you comment on that?
Yeah, I think radical polymerization again can influence cell viability. I think what you see is that obviously it's been very successful. There's a broad range of different photoinitiators also used. I've only mentioned a few today, and there's a number of combination, and I've mentioned some in the UV range, some in the visible light range, and then there's also a range of other different chemical cross-linking approaches. So I think one of the advantages, I guess, at least with the ruthenium SPS system, is again around the efficiency. So again, that high molar absorptivity is that where we know that we have the greatest potential to do cell damage is if we have to use really high light intensities and really long exposure times. That's where we beat these cells over the head quite a bit. And so again, if we can have things that can rapidly cross-link in the presence of very, very low photoinitiator concentrations using low or very short exposure times, then that's when we give ourselves the best possible chance. So I hope some of the examples I've shown today mean that again by tweaking or tuning some of those different photo cross-linking conditions, you can see that you can get quite good penetration depths or fabricate quite large constructs without necessarily needing to use very high concentrations of photoinitiators to make sure that you get complete cross-linking, or that again you can still make large constructs within relatively short periods of time. But yeah, I mean, some of the work then you've done and others around the volumetric printing is also a real advantage, that you can use those photoinitiators but make really, really large constructs in very short periods of time. So those are really nice strategies to make sure that you have to use essentially photoinitiator somewhere along the way if you go down that route, if you want to use light, or that's your cross-linking mechanism. But I think obviously those are some nice strategies to make sure that you can get what you want, but then also your cells are as happy as possible.
Thanks, it was very clear. To remain in the realm of photopolymerization, there's also a question from Abhishek Dand. Great presentation. And then he's asking about your gel age system. If in addition to tyrosine step growth polymerization, you also see competitive chain growth due to reaction between the different allele groups. And if yes, how can you distinguish the tyrosine from the allele cross-linking?
I think that's a really good question. And I think we tried to investigate it as much as possible in terms of yeah, in terms of the different cross-linking mechanisms, and it's totally right in that you know, there's a range that we can try, a range of different mechanisms or mechanistic approach which can be occurring there. So I think what I could do is try to point to the advanced materials paper that we published to then get some further information. But I think, yeah, the short answer is that I think yeah, there's still a, you know, there's a broad range of potential opportunities in amongst all of that, and I guess we've tried to investigate it as much as possible, and we've seen that the system that we have at the moment again is working well, and that we can get heterogeneous properties, and again, tailoring what we also want to try and do further with a range of different types of cross-linkers are able to help us control that a bit further. But I think, yeah, there's still further work that we'd like to be able to try and investigate, but in our hands, it seems to be working as a very tunable system at the moment.
Okay, thanks. Shrek, I will leave the last questions to you.
Sure. So I guess maybe 2 more questions before we wrap up due to time issues. So the first one is by Rios, actually. So he was saying or asking, you have talked about both spheroid and single cell embedding the gel for cartridge repair. And like when translating that to clinical research, do you have a preference between one of them?
No, good question. Thanks, Josh. So I guess there's a number of different preferences. Obviously, I personally quite like using the cell encapsulated hydrogels, mostly because, again, we can generate spheroids very, very rapidly, and it might mean for a single surgery approach that we could also take cells and encapsulate them and potentially inject them or bioassemble them rapidly. That's an area where I think is an advantage. I think there's still some further work in terms of exactly which is the best possible material. Again, whether it's going to be gelatin based or not. Obviously, if we could include, or again, the cells were able to only pure cellular modules and fabricate or look at generating high quality tissue without requiring any biomaterial hydrogel, that would be preferable. But again, to do that process, it means we need extremely high cell numbers, potentially looking at co-culture systems or the use of allogeneic cell sources to make sure we would have enough cells. One advantage of using cell-laden or the microgels is that we can provide a good microenvironment, but potentially mean we don't need as high a density cell numbers as we would potentially need with just straight cellular spheroids. So it comes down to in performance. We're investigating both, and I think that's where in future where we're working towards in our translational models is to try and test again which is going to give us the best outcome. And I think it's always going to be a bit of a trade-off between surgical complexity and potentially how much time it might take versus simplifying the process. And that's where I think the microgels or cell-laden microgels may have an advantage in terms of simplifying the surgical process.
Cool, thanks. And the final question is actually quite related. So the question was about throughput. So how do you actually address the scalability challenge in order to produce clinically relevant implants for spheroid-based implants?
So yeah, I think obviously what I was trying to show is that the microgels, if we can take our cells, so again, these could be again primary cells or mixed in with autologous MSCs or allogeneic MSCs, for example. But obviously, if we can take our cell combinations and that we can then form large numbers of spheroids in high throughput. So we showed one format where, again, we had the 96 well plate format. So the idea is potentially for clinically relevant applications, if we see advantages in bottom-up assembly, is that you could generate hundreds or potentially thousands of spheroid-based procedures. Of course, the trick is how can you then automate the assembly? I showed you sort of one example where we had we're able to automate the process of singularization and assembly to generate a large construct. So I think the microgel techniques, again, because it's a microfluidic based system, means you can generate hundreds of spheroids within minutes. Again, now if we can automate the process, then you might say that for a clinically relevant construct, example for a cartilage repair, we might be looking at say around 20 millimeters in diameter, 5 millimeters thick, or potentially a little bit more. That within half an hour you can have an automated assembly process. So again, if you can have strategies which can isolate cells rapidly, you can incorporate them into the fabrication process, then that's quite attractive. It's a different story if you then have to, you're wanting to form spheroids, or they need to mature over time in terms of the self-assembly approach. Obviously there potentially is other strategies or other tissue repair strategies where, being able to take say a one week tissue culture or maturation process prior to implantation may be an advantage. So again, that's where there'd be different trade-offs in terms of whether you'd like to have something scaled for a single surgery versus whether you've got some capability for looking at a longer term culture process. Either way, I hope I've presented at least some examples where each of the different steps has essentially an automated capability to it, and still provides I guess a microenvironment or a niche where we can still get functional tissue, or at least of each of your individual modules, which as a whole can come together to generate a more functional overall construct that's clinically relevant sized.
Thanks, thanks a lot, Tim. Thanks, everyone, for joining the webinar. Thank you for all that have joined all of the series and all of our invited speakers for the whole year as well. This is our last webinar for 2020. There are a number of recordings that we have uploaded onto the ISPF membership portal. So if you're a member, you should have access to them. And also stay tuned for more initiatives coming from us for next year as part of ISPF. And we're also going to have a Twitter poster conference that we're going to run in February, and we will be putting out a lot more information in the near future. So thank you all. Thanks for joining us today, and have a great day.
Can I have one very final thing? I'd just like to, on behalf of everyone, thank Koen, Ricardo, Shrike, and Andy for running these series. I don't know if anyone's necessarily had a chance to thank you, but you guys have done an amazing job. So thanks very much on behalf of ISPF also for all your initiatives. And I'm sure everyone else there would also be clapping. So I'd like to say a big thanks, and thanks to you for all your hard work this year.
Thank you very much.
Bye, everyone.